This conference will now be recorded.
We'll be able to go over those at the end. And then lastly, I want to let you know we are recording this session, and a link to these recordings and a link to an evaluation of today's conference will all be emailed to all the participants next week. So before we get started, I'd like to thank today's sponsors: Bayada Healthcare, New Motion Foundation, Maxim Healthcare, Ability Center, Permobile Foundation, and United Access. And we're going to take a moment to play a brief video from Quantum Rehab. And I think I'm having problems with the sound. So if the sound isn't coming through for you, please be patient. We'll go ahead and move past this, and then I'll figure that out a little bit. So. Was anybody able to hear that? Can you shake? Oh, perfect. It worked out. I'm impressed. Thank you. Okay, so thank you to Quantum Rehab. Now let's go ahead and get started. I have the honor of introducing you to our first presenter, Dr. Robert Bowser. Dr. Bowser is the John and Betty Vandenberg Chair of Neuromuscular Disease and Professor and Chair of the Department of Neurobiology at Barrow Neurological Institute and Dignity Health Medical Center in Phoenix. His research focuses on the discovery and use of biomarkers to uncover mechanisms of neurodegeneration associated with ALS, translating those findings towards new therapies, and the use of biomarkers in drug development and clinical trials. Dr. Bowser co-chairs the Northeast ALS Consortium Biofluid Repository, or NEALS, and is a member of the Target ALS Postmortem Tissue Core, overseeing a large national biofluid and tissue repository of samples collected and distributed to scientific investigators. Thank you for joining us today, Dr. Bowser. And I'm going to switch over to the other slide here.
Thank you very much, Amy. And I just want to thank everyone for participating today, getting up nice and early. At least you're doing it from your own home, so you didn't have to drive somewhere and get somewhere. So at least that's a bit of a plus, though there's always technical challenges with all of these presentations. And one of ours was hopping on at the last second and trying to get the Go meeting to work on my computer. And so right now, Amy's got my slides and will have to forward things for me since I couldn't get it to work on the laptop I was trying to work on at home. So again, thanks. Just a brief overview. Amy already introduced me. So next slide. First, I just want to mention there's been, obviously everyone's dealing with the COVID pandemic. COVID has definitely impacted ALS research nationally and internationally. Several of the sort of state and national academic medical centers were closed during the pandemic, some of which have only very recently reopened their doors for research purposes. So that has slowed down research efforts and negatively impacted a lot of collaborative research projects. So even some of mine, my lab, we remained open at the Barrow. We just had a reduced workforce within the labs, but many of my collaborators around the country were completely shut down. And so a lot of projects were stopped. And so there's been a bit of a pause because of that in research in general and in ALS research. Oops. Amy, what'd you do? There we go. I mean, another big issue is obviously patients weren't coming into the clinic. that halted a number of clinical research studies that relied on patient visits for recruitment and testing on site. New clinical trials were halted, couldn't initiate, couldn't enroll. Obviously, due to the nature of the COVID-19 virus, vital capacity, forced vital measures weren't being done in the clinic, so people weren't exhaling hard. And so a lot of things were very challenging. There was a lot of pivoting that went into how health is provided to ALS patients, and I'm sure everyone on this line has participated in telehealth approaches now for at-home healthcare, and so that is something that will continue. It's not going to go away now, and so now I think as we move forward, a lot of things are just changing, and we have a changed environment now in research. And finally, a lot of long-term experiments. So my lab, other labs, a lot of collaborators using patient-derived stem cells that often take months to grow in order to do one experiment. They were just halted because we couldn't have people in the lab on a day-to-day basis in order to maintain those cells. So many different types of experiments then were aborted because of the pandemic. Next slide. So that was an overview of some of the problems and challenges that we've all been facing due to COVID. Even prior and continuing through that, ongoing problems in the space of ALS research obviously are defining patient subpopulations that we hope might best respond to specific treatments. Obviously, ALS is a very heterogeneous disease. There's fast versus slow progressors. There's very complex genetics, over 30 genes involved in ALS. You have environmental risk factors, genetic risk factors, and then if you look at the underlying molecular mechanisms within the disease, there's a multitude of those, and some of the initiating factors then probably differ between individuals. And so it's not surprising that any one drug may not be the magic bullet for this disease. And as we've been moving forward over the last number of years, the idea is can we identify the right types of patient populations to treat with the right types of drugs? So one of the solutions for that is using biomarkers to help define what patient populations to treat with what drugs. And as Amy mentioned, my lab's been involved in identifying biomarkers now for probably at least 15 years or so for ALS. And so we've been at the forefront of identifying biomarkers and try to utilizing them within research studies and clinical trials. Next slide. So as I set out many years ago now, looking for ALS biomarkers, some of the goals that I wanted for these markers were, we could maybe help reduce the time for diagnosis. Sometimes it's challenging to identify who has ALS and who doesn't, and could we enable clinicians to make a more rapid and definitive diagnosis? That would help get people into trials very early after symptom onset. We wanted to use markers to identify new drug targets, new insights into the disease mechanisms, biomarkers to demonstrate that a drug that's being used in a clinical trial is actually doing something in the patient. Is it impacting the pathway, the biological pathway that we think that drug actually targets? So can we identify and demonstrate target engagement? Is it doing something in the patient? That can help shorten trials. Because if we start a trial and find it's not actually hitting its target, it's not doing what we think it should be doing, we can halt the study. Can we develop smarter clinical trial designs to enrich in patient populations, therefore selecting those that might best respond to the treatment? That'll reduce the number of patients required to determine whether or not that drug is having an impact. And then also identifying these sort of different ALS phenotypes. Obviously a fast and slow, bulbar versus limb, lots of different genetic variants. And all of these types of ways to identify these patient populations, if it's genetics, if it's different proteins, are all biomarkers for the disease. Next. Oh, so that last little blurb, I forgot, there's still a couple, I guess, I thought I took off. Yeah, so really the goal was to develop sort of a biomarker toolkit that we can use in research and clinical trials. Next slide. So I'm going to talk about a couple of types of biomarkers that we and others have been looking at, and providing some additional, very near-term, very recent study results that we've been performing in the last year. So the first group are biomarkers for inflammation and modulating the immune system. And a lot of different ALS clinical trials are really targeting neuroinflammation in the immune system. We know that inflammation is activated in almost every ALS patient, but the immune system is quite complex. And so you can have different types of immune responses in different patients. And so can we identify biomarkers for different types and pathways within the immune system, and take the growing number of drugs that are targeting inflammation in the immune system and really use those in selected patient populations. So the prior clinical trials in ALS that have really used some of the drugs that target inflammation, but in a very general and broad sense, all failed. And so we have to get smarter, and we have to get fine-tune what pathways these drugs are actually impacting and targeting. A number of different types of proteins that are called chemokines, cytokines, which are inflammatory proteins, have all been looked at as potential biomarker candidates for ALS. Many of them are increased in ALS patients versus control groups, either in the cerebral spinal fluid or CSF, or in the blood. Next slide. So here's some data that we published just recently. I think this paper came out a couple of months ago. And this is a collaborative project with my lab, James Berry at Mass General, and then a group headed by Carol Ho at Denali Therapeutics. And so they're very interested in targeting the inflammatory pathway, and we were looking for determining what types of inflammatory cytokines might be useful for their purposes in drug development. And so this is just 3 of these different types of cytokines. And on the top 3 panels are levels in cerebral spinal fluid, and the bottom are levels in plasma. And what you can see, which is the controls, are sort of the dark circles, and the somewhat goldish or yellow is ALS patients, that there's elevations in these particular cytokines, especially in the CSF. And if you look in the blood, maybe it works for MCP1 and soluble TREM2, but maybe not so much for IL-18. So maybe IL-18 is more specific for CSF and not blood. So sort of click on it. I think there's an animation that, oh no, never mind, I'm sorry. So really what we found here is particular cytokines that are elevated and changed in ALS patients versus controls. And now, next slide, if you look at different types of subsets of patients, and now we took that same patient group and we looked at patients that were slow progressors, which are in this case the sort of yellow gold color, fast progressors, which are somewhat orange, and patients that harbor the C9 ORF72 repeat expansion, which is in red. And so up on the top panels, again, looking at CSF, you see that a number of these, like MCP1, IL-18, and soluble TREM2, are actually at their highest levels in patients that are either fast progressors or harbor that specific C9 repeat expansion. And in the blood, you can see somewhat similar results, but actually it changes in some. So for instance, IL-15, which is the third in the bottom panel, actually goes down when compared to controls in, say, C9 patients. And so we start to separate and start to identify specific, in this case, inflammatory cytokines that appear different in different subpopulations of ALS patients. And so we can use that to our advantage in research studies and clinical trials that will be upcoming. Next slide. Another group of proteins that my lab and a number of other labs have been studying in ALS patients are proteins called neurofilaments. And there are sort of two flavors. There's neurofilament heavy chain, which is sort of abbreviated NFH, pNFH, and neurofilament light chain, which is NFL. And these are proteins that are only made in neurons within the nervous system. And when the neurons are damaged or if they die, it's released outside of the cells. That accumulates in the CSF and ultimately gets drained into the blood. And so we can look for the presence of these neurofilament proteins as a biomarker for damage or death of neurons within the nervous system. And so here's just an example again from that recent paper showing that in the CSF or in the blood, we see elevated levels of neurofilaments when compared to controls, and again, the highest levels in either fast progressors or patients that harbor the C9 repeat expansion. So the neurofilament proteins are sort of one of the top biochemical biomarkers for ALS. And a number of groups, including my own, have been using these as markers to follow patients over time. They stay fairly constant over time. And that's good because our interest is trying to see a decrease in neurofilament levels in response to a drug treatment. Next slide. Here was a paper published at the end of 2019 between my lab and Michael Benatar down at the University of Miami. And Michael has been, for a number of years, been following patients in a longitudinal manner. And these patients have mutations that are known to cause ALS, but they are asymptomatic. And Michael's been following them and collecting blood and some CSF samples and following them over time and saying, well, when do they first develop symptoms for ALS and now convert and become an ALS patient? And then can we look backwards in time and say, do any of the biomarkers of interest actually exhibit alterations or increases even before symptom onset? So can we identify biomarkers that actually might be altered in patients or or individuals, but even before symptoms occur? And the neurofilaments are the first of these proteins that we identified that actually appear elevated in a number, not all individuals, but a number of individuals that ultimately then are diagnosed with ALS, but before symptom onset. And so in this figure, the sort of 0 and the elevated vertical bar, that's the time in which they were diagnosed. They had symptoms and now could be diagnosed with ALS. You see a number of individuals that for up to even 1, 2, or even 3 years prior to symptom onset, there's an elevation in neurofilament within either their blood or CSF. And so this was very exciting. It's the first study showing these elevations of particular proteins in people before symptom onset. Next slide. And here's a great example of how these become very valuable. This is a different disease. This is spinal muscular atrophy. And this is in children with SMA. So in this patient population, the SMA gene, there's alterations and they make less SMA protein. And very bad disease. It's often called floppy child syndrome. So they're infants, they can't control their muscles. Ultimately, the individuals pass away. For a number of years, groups have been looking for treatments. This is a particular phase 3 trial of a drug called Nusinersen, which modulates the expression of the gene within individuals to make more protein. And so in this case, what we're showing here was results from a phase 3 trial where they're looking at neurofilament levels in the blood from these babies that were either treated or not treated with the drug. And in red, what you see is this tremendous decrease and rapid decrease in neurofilament protein in patients that were treated, children that were treated. All of these children had basically a reversal of their symptoms, and now many of them are walking again. And so we had a correlation between a drop in this biomarker in the blood and a change in the clinical parameters and improvement in the patients. And so this was, in the neuroscience space, one of the first examples of using these biomarkers and demonstrating that, yeah, if we can actually impact and, in this case, reduce the level of a biomarker in the blood, that correlates with a change in the clinical parameters of the individual and an improvement, in this case, of the children with SMA. Next slide. So again, very exciting results. This is a different set of proteins that are back into sort of the inflammation pathway. They're called chitinase proteins. I was involved in studies back about 15 years ago when we first identified these in the cerebral spinal fluid. And there's a small table from a publication from 5 years ago from my lab showing, again, increased levels in ALS patients versus, in this case, healthy controls. And these proteins are expressed by immune cells within the nervous system that modulate both the innate and adaptive immune responses, remodel all the sort of space in which the neurons and the other cells within the nervous system live, and also can promote sort of vascular regeneration and blood vessel generation. Not a whole lot is known about these, but since they're secreted molecules, they can actually be used quite well as markers. So there's 3 different proteins. We've been studying 2 of them more specifically. The first one called CHIT1, which was known to be expressed in activated microglia within the brain. The second one, just abbreviated L1, which is expressed in different types of immune cells, either astrocytes or microglia. And so we are interested to see and further study these in ALS patients. Next slide. So this is data from a paper that came out earlier this year from my group, where we are looking again at either CSF or blood levels of different chitinase proteins between ALS patients, other neurologic disease controls, abbreviated DC, or healthy controls. So on the top is CSF, and CHIT1 levels were dramatically increased in ALS patients versus healthy controls, but also against other neurologic diseases that mimic ALS. L1 was elevated in both ALS and the disease controls when compared to the healthy controls. And interestingly, when we look in the blood from these same individuals, we really didn't see any significant changes. So suggesting that sort of the peripheral and the central immune responses are different in individuals during ALS. So what's happening in the periphery, at least with respect to this particular type of proteins involved in the immune system, versus what's happening within the brain and the spinal cord, the central nervous system, differs. And so it's very interesting. We don't know why that's true, but we're interested to see how some of these proteins might be actually trafficking back and forth between the periphery, and that is the blood, and the central nervous system. Next slide. We use some of our postmortem samples. So as Amy mentioned, I run a big tissue banking program where we collect postmortem tissues from patients and controls. And we asked, for L1, where's it actually expressed? What cells are making it in the nervous system? And this is just one of the figures from this paper showing that L1 is actually expressed in activated astrocytes, another type of immune cell, predominantly within the white matter. So not throughout the whole brain and spinal cord, but only in specific locations. So it's subsets of these astrocytes and predominantly in the white matter. We think it might be contributing to the ongoing degeneration of the axons, the fibers that are coming from the motor neurons in the cortex and going down to the spinal cord, and being involved in modulating the injury of those axons in ALS patients. So it becomes a target for additional drug therapy development. Next slide. This is data from actually a collaborator, Martin Turner's group at Oxford in the UK. And towards the end of last year, he published a similar study looking at chitinase proteins and their levels. And this is, again, just looking at the CSF. very similar results that we got, elevated in ALS versus a lot of other control groups. I included this panel only because on the far right for each of these little graphs, there's a group called AGC, which are actually asymptomatic genetic mutation carriers. So I showed you some data saying that neurofilaments actually increased even before symptom onset in this patient population that harbors genetic mutations. Doesn't look like the chitinases in the CSF are. So they remain low before symptom onset occurs. So again, a nice distinguishing feature between certain biomarkers. Next slide. So some summary of these results. So we know a number of different biomarkers for the inflammatory system that exhibit significant changes in the blood or CSF of ALS patients. And we can start to distinguish between certain subgroups of ALS patients, either easy ones or fast versus slow, but also different genetic subtypes. And that becomes very important as we move forward into different types of clinical trials with specific drugs that target the immune system. Other types of inflammatory proteins, the chitinases, are really only altered in the CSF. But they can also distinguish different types of immune cell activation. So as I mentioned, L1 was only in these cells called astrocytes. And so if the drug targets astrocytes, this might be a great biomarker to use. But if the drug targets other cell types within the immune system, this probably wouldn't be a good biomarker to use. Finally, neurofilament proteins are definitely elevated in the blood and CSF in ALS patients, and in some individuals even prior to symptom onset. Showed you some data from an SMA phase 3 trial that was published last year, demonstrating that drug reduced levels of neurofilament, and that correlated to clinical improvement in the patients. And so it becomes a great biomarker to continue to explore in ALS clinical trials. I'm going to shift for a moment and show you some data from another study we've been doing in a completely different area of the nervous system that, especially in ALS, has really been ignored throughout all these years. And it's a region called the choroid plexus. And so in this image here in the slide, you see what is shown. Too bad I don't have my marker I can show you, but the choroid plexus, which is somewhat, oh, there you go. Yeah. So that color isn't real. We sort of put in the color just so you can see it easy in comparison to the rest of the brain tissue. But the choroid plexus it's a very thin layer of cells, and it lines what are called the ventricles within the nervous system. And two of them are shown here. So these are the lateral ventricles. And so you have this thin layer of cells that lines these ventricles within the nervous system. So what does this layer of cells do? Well, first off, it makes all the CSF in your nervous system. So it's constantly churning out CSF, and it creates that because it's highly vascularized. That means it has a tremendous amount of blood flow through this tiny thin layers of cells, and it basically creates plasma out of your blood, has a group of cells that adds other proteins to it, and it sort of creates CSF. And it constantly does that. Listed there at about 500 mils a day throughout your life, because there's a constant turnover of CSF multiple times every day throughout all our lives. So this is a very important little layer of cells. It's also an interesting area because it's one of the gatekeepers of other cells from the blood entering into the nervous system. And in a couple of different model systems, animal model systems, there are tremendous changes of the choroid plexus, mainly in other diseases like Alzheimer's disease and Parkinson's disease. A number of years ago, some of the biomarkers that we identified in CSF are those that are actually made from the choroid plexus. So we got interested in studying the choroid plexus, yet no one was studying it. No one had any tissue. And so we basically had to create our own a bank of choroid plexus tissue that we are collecting from our tissue bank in order to do any studies. So it took a few years to generate a bank of choroid plexus tissue. I used other collaborators around the country and to collect this tissue, but we've basically done the first and definitely the biggest, one of the biggest choroid plexus studies for a neurodegenerative disease. So next slide. So this is just showing the name of the publication. It came out about 2 months ago. And as you can read, global alterations of the choroid plexus, this blood CSF barrier, important barrier within ALS. Next slide. So a couple of pictures. So these are images of this thin cell layer, sort of cut cross-sectional. And the top two panels are control individuals. And the bottom two are patients that passed from ALS. This is looking for one of those biomarkers that we had seen in the CSF. It's some protein called fibrinogen, and it's a clotting factor actually, and it's made in platelets. And obviously in the red on the bottom, you can see a drastic increase in fibrinogen that lines these cell layers within the choroid plexus. You don't really see this in the brain, but you see it in the choroid plexus. And so there's platelets here that are dumping out fibrinogen within the choroid plexus. Next slide. A couple of more images here showing, again, I mentioned it's a gatekeeper. And so some of the cells, immune cells that are in the blood, in the periphery, are moving into the central nervous system through the choroid plexus. And sort of the left three panels are from a control individuals, and on the right are ALS patients. And using different markers, you can see an increase within the choroid plexus of ALS patients of these immune cells that are migrating from the blood into the central nervous system through the choroid plexus. Next slide. And so we discovered this sort of widespread structural and functional disruption of the choroid plexus in ALS patients. It's never been looked at. No one ever knew it existed. We see an increased number of immune cells crossing into the nervous system through this area during ALS. So we've identified the choroid plexus as a brand new target for therapy and drug development. Can we stop those immune systems from those immune cells from going into the CNS through the choroid plexus? Can we target the changes and disruptions of this really important cell layer that occurs in patients, and what would be the outcome for that for individuals? Can we modulate progression, slow disease progression by only just targeting the choroid plexus within patients? And the nice thing is, since it is highly vascularized and is the gatekeeper with the blood, we could easily target it by just providing drug treatments right into the blood. We don't have to go into the CNS. Next slide. So that was very exciting. Now, I just mentioned one of our challenges in drug development for ALS and other neurodegenerative diseases is that it's often hard to get drugs into the central nervous system. Most drugs don't cross the blood-brain barrier, which is good. That's why that barrier is there. It's to protect the nervous system. So we always need and looking for better ways to get drugs into the nervous system. One approach, which has been used a lot lately, is basically, well, let's just put the drugs directly into the CSF using an intrathecal delivery method. Obviously, it's an invasive approach, and you can either inject multiple times, so have the patient come into the clinic multiple times, inject directly into the spinal canal, or sometimes you use pumps. And you basically put your drug in the pump, put the pump underneath the skin of an individual, and then basically have a tube from the pump going into the central canal, and then the pump slowly delivers drug constantly into the intrathecal space. And then you just have to change the pump with more and more drug every month or so. And so working with some bioengineers and one of the fellows in my group, we looked at other ways that we could try to package and target delivery of drugs into the central nervous system. Next slide. So what we did was we developed what's called nanoparticles. So very small little lipid bilayers, so small little circles. So that in the middle are some pictures of these nanoparticles that we create in the lab. And we created them very special. So we put ways to try to target it into the central nervous system. And then we could put drugs inside these nanoparticles. So basically encapsulate drugs into the particles. And the first one that we did was this actually a retinoid molecule called adapalene, which is FDA approved. It's actually a topical agent. And so people administer it to the skin. It doesn't cross and doesn't get into your bloodstream. If you put it in the bloodstream, it never gets into the brain. And so we had data showing that these retinoids are actually very neuroprotective for motor neurons, but we can never get the drug into the brain. And so what we decided to do is ask, well, can we put this retinoid into these nanoparticles and then test them in the SOD1 mouse model of ALS? Next slide. So we published this paper earlier this year, and basically, again, showing that we could take these nanoparticles, put them into the bloodstream of mice, and show that it activates retinoid signaling in the central nervous system in these mice, and it increased the lifespan and reduced motor neuron loss within this mouse model of ALS. Next slide. So just a little bit of data from that paper. The top is looking at survival and progression. The ALS mouse model, as some of you might be aware, is a very aggressive form of the disease. The only way to really cure the mice is to basically turn off the expression of SOD1 in the mice. So this is just showing in the blue, again, there's an increased survival, which for drugs that are successful in this mouse model, that's kind of very similar to the curve one gets. And again, with progression, it's slowing the onset of the disease within the mouse model. And in the bottom, we're just showing with some bar graphs showing that we're counting motor neurons in these animals over time. And the ones that are sort of decreasing rapidly over time, those are animals that were injected with empty nanoparticles, controlled nanoparticles, and they're losing motor neurons over time. And the lighter colored bars are the animals that were treated with the nanoparticles containing the retinoid, and these were retaining motor neurons over time. And the right were just a couple of images from either animals at the end stage that were either treated with the control nanoparticles on the left, and then looking for a marker for activated microglia, so you can just see lots of little red dots. And then in the animals treated with the nanoparticles containing this drug, there's fewer of these dots. And so we've reduced inflammation within the spinal cord using these nanoparticle drug delivery methods. Next slide. So again, very exciting. It's a new way to get drugs into the central nervous system. So we've demonstrated we can package these types of drugs that normally can't get into the nervous system. And we can effectively target them into the central nervous system. And with the retinoid that we use, we can reduce neurodegeneration and reduce inflammation in the mouse model of ALS. And we're still many steps away from bringing this type of therapy into human clinical trials, but we are pursuing that, and right now working with a couple of different drug companies because we have to be able to take sort of the creation of these nanoparticles out of our lab and then put it into a facility that could create nanoparticles under the right conditions that via the FDA that would permit them to be injected into people. And so we are pursuing this line of investigation. So hopefully we'll be able to try to test these types of nanoparticle drug delivery approaches, and I would guess it's probably going to take about 2 years still, unfortunately. Next slide. Now I'm just going to go over in the last 15 minutes or so some recent drug trials that have come out. First group are trying to reduce SOD1 in patients that harbor mutations in SOD1. And there were two approaches. You probably heard at least of the first one, which one was from Biogen, that were using what's called antisense oligonucleotides, ASOs, and their particular ASO for SOD1, the trade name for that is Tofersen. And so they were taking these antisense oligonucleotides, provided them to patients that harbor genetic mutations in SOD1, and they wanted to see what type of impact it had on those individuals. The second one there is something called AAV viral vectors. So AAV stands for adeno-associated virus. And so this is a viral vector approach. That SMA trial that I showed you many slides ago, showing the great impact on neurofilament, that was an AAV vector approach of drug delivery. So it's a gene therapy approach. And again, the approach by using this viral vector was again to modulate SOD1 gene expression in patients. Next slide. So this is data from a paper from the New England Journal of Medicine that came out July 9th, so a little over a month ago. So this is again, data from the Biogen study. They are delivering their ASO, their antisense oligonucleotide, to modulate SOD1 expression using this intrathecal delivery approach that I mentioned. And so essentially it's multiple injections into the spinal canal of the people that are in this trial. So the top three bar graphs there, or not bar graphs, but line graphs. On the left side, it's ALS-FRS score. And there's a black line going over the top, which would be no change in the individuals. The blue line is placebo, and the gold line is the drug at 100 milligram dose for each injection. And so what they noticed, again, if you just look, it's now again up at the upper left, is that there's, this is only in days, so 85 days later, there is a modest change or a small change between the placebo group and this is the high drug dose group. If you looked at vital capacity in the middle, again, maybe a change, maybe a slowing in the reduction over time of vital capacity measures. And if you look at strength measures, which is in the right handheld dynamometry, again, patients given the drug, there's a little blip up, and then 90 days later, there's really no change over time. Where with the placebo group, there's a small change in reduction of strength measures over time. Again, short study. This is only 92 days after dosing. What they did next was they said, all right, well, let's try to look at subgroups. Let's look at fast progressors in this case. And so again, it's only a subgroup of the patients. Now it's only N of 4 in the placebo and the drug treatment group. But now if you look at fast progressors, over the course of this 85-90 days, on the bottom left is now ALS-FRS measures. And you can see a change over time in the fast progressors. But the fast progressors that are treated with the drug, there's no change over time. Highly significant. The middle panel, again, is looking at vital capacity. And again, there's a shift, there's a change, there's a slowing of the loss or progression of vital capacity measures. And again, on the right, looking at strength measures, again, you see that sort of blip up in the drug treatment group and the fast progressors. And then over time, no real change. It goes back down to baseline. Whereas the fast progressors placebo group, you can see the loss of muscle strength over time. And so when they looked at the subgroup analysis, they were very excited. And so it looked like the drug was doing something in this patient population. Next slide. They also wanted to look at SOD1 levels. And so if their drug was reducing the amount of SOD1 actually accumulates in the cerebral spinal fluid. And so they could measure SOD1 protein levels in the CSF. And so if the ASO is working, then you should see a reduction. And sure enough, in the 40, the 60, and the 100 milligram doses, so the black, the yellowish, and the goldish or orangish, again, one sees a reduction over time in the amount of SOD1 that's in the CSF. And so again, it's demonstrating that, yeah, likely the drug is hitting its target and it's doing what it should be doing, which should be reducing SOD1 levels. Next slide. And so now again, here's another biomarker. So now they went back and looked at neurofilament levels. And similarly, like the SMA drug trial, could you see an impact of this antisense oligo in neurofilament levels, which is a marker of neuronal injury and death? And sure enough, with either the heavy chain or the light chain, looking at blood levels of neurofilament in response to drug, the higher the dose, the lower the levels of neurofilament in the blood of patients over time. And so this is a very promising result. And again, a great indication that for this patient population, for patients that have SOD1 mutations, this might be a very good drug treatment for this patient population. So they've gone to the FDA with these results, even trying to get approval now, but at the same time, concurrently, they're running a larger phase study with more patients. So the total number of patients in this study, I think, were 50. And so they're increasing the number of patients and repeating at just a higher dose for the patient population. So great results. The challenge here is, as I mentioned, it's an intrathecal delivery. And so that means the patients have to go back into the clinic every so often and get injected into the spinal canal with more and more of this antisense oligonucleotide. Next slide. So this was the first effort of trying to use this AAV, these adenoviral vectors, to modulate SOD1 mutations in patients that have the SOD1 mutation. So basically, again, similar to the antisense, trying to turn off SOD1 production in patients that have this mutation. So again, this was a paper that just came out. It was basically the companion paper to the one we just went over. This, instead of a big drug company like Biogen, this study was run by an individual, Bob Brown at the University of Massachusetts, an actual well-renowned ALS clinician and geneticist. And so he's been working on this for a couple of years, created these vectors, tried it in 2 patients. Again, it was really more of a safety study than anything. Can we deliver these AAV vectors into an ALS patient and make sure it's safe? So it was only 2 patients, and the top panels here are just looking at vital capacity and ALS-FRS. And what you see is in patient 1, which is in the red, the patient continues to decline over time. And in patient 2, which is in the blue, very likely a very slow progressor, there's really no change. So from the clinical parameters, you really can't say that this treatment did anything. But again, N of 2 type of study. The patient number 1, who progressed very rapidly, passed away, and they did an autopsy on the patient and collected tissues. So the bottom panel is images from the spinal cord from the individual, showing that on the left, the lower left, trying to look for motor neurons, you really don't see any motor neurons there. And on the right, you do see motor neurons surviving. And there's sort of the sort of big purplish type of cells that you can see. There's about 1, 2, 3, 4, 5 of them. And so what he noticed was that in this one patient, on one side of the spinal cord, there was a number of motor neurons throughout the spinal cord. So it's sort of that it was protecting the motor neurons. But for whatever reason, on the other side of the spinal cord, there's no protection. So it could be that this virus that was injected into the patient was sort of suboptimal delivery. It only hit some areas. It only hit some motor neurons. The nice thing about this type of an approach, if it would work, is that it's a one-time treatment. So instead of going back for injections, intrathecal injections, every couple of months for the rest of your life, this is a one-time treatment. You go in, you get one injection, and you're done. So if it works, it has a lot of advantages. But again, very early study, very early phases, and really all it was showing was that it didn't do any harm to the patient, and again, really did not have any of the dramatic changes or clinical parameters that was seen in the Biogen study. Next slide. So the next trial is a study from Amylyx, which is called the Centaur study, which is actually a combination of two drugs, both of which has been tried in ALS patients in the past and both failed. But they had an idea that if we combine these two drugs together, we might see a slowing of disease progression. Next slide. So really, they're just reporting on publishing the outcomes. But what they have mentioned in a few different public sort of media releases is that their primary outcomes were met. So what they saw in patients that were treated with the drug versus placebo was that there was a slowing of basically ALSFRS functional measures. So it impacted speech, swallowing, general areas of dressing and hygiene when compared to those on placebo. The biomarker data is still being sort of explored, and my group is involved in that data analysis. And so that's an upcoming portion of the study. But at least the initial indications were that in this study of over 100 patients, that there seemed to be some clinical measures that were impacted by this drug. Again, very exciting and worthy of continued pursuit. The next study is a drug called Actemra, which is a drug, it's a humanized monoclonal antibody that block an inflammatory pathway, IL-6 signaling. Next slide. So this study was run by Shafiq Lada at the Barrow. So it was a phase 2 study using this drug, humanized monoclonal antibody, tocilizumab, in ALS patients, and it was a multi-site study. So it included both patients at the Barrow, at Wake Forest, at Penn State, at Mass General, and at the University of Kansas. Next slide. I've gone over neuroinflammation and ALS, lots of biomarkers, lots of indications that yes, inflammation has changed in many ALS patients, but again, it's not always the same in all patients. This drug only targets IL-6 signaling, and it blocks that signaling, and so we were interested in looking for patients that had altered IL-6 signaling, and those were the patients that we were going to target for therapy. Next slide. So the study design, I won't go through completely from the study design, but really the important thing was up on the top, we screened a number of patients, and if you had what we called low inflammation for IL-6 signaling, you were excluded from the study. There is no reason to include you since we know the drug targets IL-6. If you didn't have that elevated IL-6 signaling, then the drug's probably not going to work in you. You might as well go into a different clinical trial. So, but if you had high inflammation, then you were enrolled in the study, and then you were separated into a placebo or a drug treatment group at sort of a 2-to-1 ratio. And so again, it was one of the early studies that we are using biomarkers to determine who should be actually enrolled in the study. It was a short study, so 16-week duration, totally only 8 weeks on drug. Again, more of a safety study, smaller safety study for ALS, making sure before we did any larger clinical trial that the drug is safe in this patient population. Next slide. But we also looked at these inflammatory cytokines that are related to IL-6 within the patients. What we saw was that when we looked at people on placebo or people on drug, we saw a significant change in this inflammatory cytokine pattern. and in the exact same direction that one would expect if the drug is having an impact. Since it's already an FDA-approved drug for other treatments, actually it's used in rheumatoid arthritis. So large amount of information around how this drug operates in humans, how it impacts IL-6 signaling in patients with rheumatoid arthritis. And just showing here, we're measuring IL-6 levels in the plasma, almost exactly opposite of what you would think. It actually increases IL-6 signaling, IL-6 protein levels in the plasma. But on the far right, what you see is this thing that's marked as soluble IL-6 receptor. And you see it also dramatically increased in patients that are receiving drug. All the plasma levels of IL-6 are actually sort of swamped up by the soluble receptor, and so basically it inactivates the protein. It can't perform its normal function, and so that's exactly the pattern that we are hoping to see. We looked at another inflammatory cytokine, C-reactive protein, and that's in the middle. And here you see that it's reduced in the plasma of patients that are receiving the drug. And so another inflammatory protein, and now this one is decreasing in exactly the same direction we were hoping to see. Next slide. This drug was given by infusion. So it was delivered directly into the bloodstream because it's an antibody-based approach. We actually took some CSF from patients and that were either on drug or on placebo. And here what you see is, again, looking at the same three markers, and you see the same pattern, that in the far left and the far right, you see this elevation in patients receiving the drug, and then looking at C-reactive protein, you see a reduction, which is that middle panel. Again, we are given this drug in the blood, and we're seeing changes exactly as we were hoping to see, mirrored in the cerebral spinal fluid. So that means that enough of this antibody is getting into the central nervous system to change IL-6 signaling within the central nervous system of the patients. We're very excited to see this. Next slide. So from this small trial, again, we're just showing safety. It was safe and well tolerated. It was too small of a study and too short of a study to look for clinical benefits, and we didn't see any. ALS-FRS, forced vital capacities, no changes. But looking at biomarkers, we saw evidence that the drug was doing exactly what we hoped to see in the patient population. And we also knew from prior studies that that individuals have genetic alterations of the IL-6 receptor. In patients with a certain genotype actually responded even better. So we're looking to move this into a larger phase study, enrolling patients that have IL-6 signaling, and ideally, if they have this genetic variant of the IL-6 receptor, that we think that people will be hyper responsive to this drug medication, and we think might reduce and slow disease progression. So again, some very exciting results, and we're just submitting this paper for publication right now. And finally, just a couple of slides on the platform trials, and you probably all heard about this. So trying to recruit 800 patients in the 5 trials. There's 54 sites across the country that are involved in this, one in Arizona, our site at the Barrow. Next slide. And really, what's the advantages of this type of platform trial? So in a traditional drug development on the left here, if we wanted to test 10 drugs, that might take around 12 years to do, typically. And we'd have to enroll about 2,400 individuals, and about half of those, 1,200, would be on placebo in order to test whether or not the drug's having an impact. So very methodically, one right after another, how long would it take to run all these drug trials? On the right is sort of the platform study, which is a very different type of trial. So here what you're doing is you're basically enrolling people into different treatment arms of the same study. What that allows you to do is it allows you to share controls. You share placebos. And so that means that within each trial, fewer people are actually on placebo, more people are on drug. And so now to take the same 10 drugs that you wanted to test, instead of 12 years, you can do it in 4 years. You would enroll 1,600 patients instead of 2,400, and only 400 would be in placebo. And so tremendous benefits of this trial design. And so again, we're very excited about the start of this. You can go to the next slide, that's fine. Because it was supposed to start in the spring. Because of COVID, we couldn't start it because no one could enroll in patients in the clinical trials because patients weren't coming in the clinics. Here are the first 5 drugs that are in this platform trial design. If you look, so on the far right, I sort of just put in a little blurb about what they might do or what they should do. And so there are a number of them, again, that are hitting the immune system, trying to modulate the immune system, looking at oxidative stress and inflammation. The third one, trying to boost up energy levels within cells and function as a neuroprotective agent. The fourth one, again, is a neuroprotective agent. And again, and the fifth one is now targeting specific immune cells, the microglia. So looking at that as a modulation of inflammation. And so lots of sort of common themes within this first 5 set of drugs that are going in the platform study. But the platform trial should be initiated rather soon. I can't give you the exact date, but it will be starting very soon. Next slide. Because of COVID, I mean, some of our challenges are, again, trying to do a lot of these measures. Can we do them at home? Jeremy Schaffner at the Barrow for the last few years has been working with a company called Aural Analytics, which is actually a company, a spin-out company from Arizona State University. That they're located in Tempe, and they have an app, and they use speech, and so they collect speech data, and then they analyze speech data on their computers and look for how speech is changing over time in patients. They have a lot of great data showing that they can see changes in ALS patients very early, and they can monitor that over time. And so most trials and clinical research studies will start using these types of at-home measures because still right now, patients aren't coming into the clinic at every visit. And so we need new approaches, and we'll be incorporating these at-home measures, I think, now always going forward. It's just what has changed in the world of ALS. Next slide. Yeah, so I mean, there's a lot of shared resources for the ALS research community. Amy mentioned about the Niels Biorepository. We're sort of the Western hub of that here in Arizona. Also at the Barrow, we run a large clinical trials training and monitoring unit. And so we run and monitor clinical trials from around the world. And then this postmortem tissue banking program that we started from Target ALS and also funding. And we have 6 sites now that are consortium members, about 200 cases now with tissue that we make available to scientists around the world. We do whole genome sequencing at the New York Genome Center. We make all this data and information publicly available. And again, we're giving out tissues to scientists around the world. Next slide. As an example, so this is just a publication that came out at the end of last year in October, using the tissue that we collected from this consortium. The groups at New York Genome Center and main group at Cold Spring Harbor in Long Island, they used the genetic information and basically said, we can identify 3 subtypes of ALS from this postmortem tissue sequencing information, and we can bin patients into different subtypes. And I'm not going to go through, you can sort of see right in the middle there, different colored. There's something called ALS-TE, which is actually transposable element. So it's actually somewhat a viral approach. There's an indication that's occurring. There's then the oxidative stress form of ALS, which is that middle portion in blue, and on the far right is the glia. So again, the immune response. And so you can start to bin people even using our postmortem tissue. And so these types of approaches are providing new and improved insights into the disease mechanisms. And again, new ways in which we look at ALS and try to stratify the patient population. Next slide. Upcoming, so we actually have an upcoming study, and if anyone's interested in participating, let me know. So this will be another longitudinal biofluid and clinical information These samples that are used by people around the world, and so we go through them rather quickly. Target ALS is sponsoring this study, and now Oral Analytics is also going to be a co-sponsor. So we're looking to enroll 280 ALS patients and 80 healthy controls. We're going to collect as best as we can during COVID, blood and CSF over 2 years. But now again, we're using these at-home measures. So we're using speech analytics, using Oral Analytics app, and we're actually going to do home spirometry measures using a company called Zephyrix. And basically, the patients will be going home with their own spirometer, and twice a month we'll have phone calls and Zoom calls with the patients, and basically be coaching them through using a home spirometer in order to get vital capacity measures. And then we're also doing whole genome sequencing on everyone with the New York Genome Center. And we're hoping to initiate this study in November. It was supposed to start last spring, but again, COVID halted everything. Next slide. Final slide, promise. Some take-home conclusions. So we have biomarkers that can really distinguish subtypes, even some that appear at or even prior to symptom onset in individuals. Really, at this point, biomarkers are being incorporated into every ALS clinical trial. You really can't move into phase 2 without a biomarker that's in the study. Antisense oligonucleotides are really one form of therapy that looks incredibly promising for patients with SOD1 mutations. But that's not the only genetic form that we think ASOs will be very helpful for. And so soon to be started trials for patients with C9 repeat expansions and for patients with FUS mutations. The AAV vectors that I showed a couple of slides on, it's a new and that's a one-time treatment approach for gene therapies. There's a lot of work to be done still, but again, it provides yet another approach for treatment for ALS. Nanoparticles, again, provide a new way to target drugs for the treatment of ALS. So the vector approach is more of a gene therapy approach one time. Nanoparticles is actually trying to move the drug into the central nervous system. The platform trials, again, will be accelerating testing of drugs in ALS patients, so incredibly exciting, and can't wait for that to continue and move forward. And lastly, I mean, a lot of some of the challenges that we've had recently are all due to COVID. As you're all well aware, it's really introduced telehealth to the world of ALS, and this will continue. So we're not going to go back to the way things were. I think it has changed the way that all trials and clinical research studies will be performed for ALS from this point on. And that's my last slide. And with that, I'm happy to try to answer any questions you might have. Thank you very much.
Thank you, Dr. Bowser. That was a wonderful presentation. There's a lot of really great news, and I was delighted and thrilled by a lot of the stuff that you presented there. We have had multiple questions typed into the chat box, and we're running a little bit short on time, so I'm going to go ahead and we'll try to get those presented and answered. The first question is...
But in for 1 sec, if we can't get through some of these, I'm happy if you send them to me, I'll answer them, and then you can send out answers to everyone.
That would be great.
So
that would be
trying to really rush through a growing number of questions, and I'm happy then to just take time and fill out the responses, and you can share them with everyone.
Okay, that's wonderful. And if we end up having to get off of here so the next presentation can start, and you had questions that we did not get addressed, feel free to email those to me. My email address is amy, A-M-Y, @alsaz.org. So if anybody wants...
Dr. Bowser?
Yes.
Dr. Bowser, can you hear me?
I can.
Okay, I'm going to go ahead and address some of the questions that were in here. The first one is, would you consider ALS to be an autoimmune disorder?
Oh, great question. To date, we don't have sufficient data to say it's an autoimmune disease. I can definitely say, I mean, my lab, a few others have tried to look for autoantibodies, and there's some data, there's some evidence that that can happen, but we don't know if that's a causative thing or if it's happening downstream. as a response to the disease. So at the moment, I would say no, it's not an autoimmune type of disorder, but there is still, the jury is still out on that question.
Okay, fantastic. Thank you. I was prescribed Riluzole. How does it help with the disease, since I assume it does not reach the nervous?
Dr. Bowser, have you seen any?
It sounds like someone's trying to present a question verbally, but we can't understand, or we can't hear it very well. So if you would just go ahead and email me that question, we'll get it presented and provide the answer afterwards.
Yeah, so Riluzole. So Riluzole can get into the central nervous system. So it does get into the central nervous system. It gets booted out, so you can't really elevate levels incredibly high in patients and taking a pill form of it. So I mean, it works by a couple of different ways, but I still think the way in which it's really impacting ALS patients is a little unclear. It's supposed to be working through glutamate excitotoxicity and trying to reduce glutamate excitotoxicity. I'm not completely convinced that's the mechanism of action for ALS patients, because it's a sort of a dirty drug and hits lots of targets. But definitely Riluzole gets into the central nervous system. It definitely impacts and it can slow disease progression. But it's only a modest slowing of disease progression now.
Okay. Thank you. And Charles, I will present your other question to him in the written questions, and we'll get those answered, because as you said, there's a growing number of questions that are popping up now here, Dr. Bowser. But I know we have time for one more. What percentage of patients with ALS have SOD1 mutations?
Yeah, SOD1 mutations, about 2%.
Okay.
So it's a small number.
Okay. Thank you for that. And then the next question, is my impression correct that today's research is focused on slowing progression versus finding a cure to allow patient normalcy?
I think we all want to halt disease progression. And the challenge is, what will it take to completely halt disease progression? I don't think any one drug will probably completely halt unless you have a genetic form in which you can probably halt the disease progression, like the SOD1 for SOD antisense. So I think that the challenge is finding groups of approaches that work in very defined patient populations. I think that will permit the best chance to finding individual drugs that will really halt progression. Ultimately, though, by finding drugs that slow disease progression, then the next step will be combinations of drugs that will further reduce and hopefully then halt disease progression. But it's not that we're not trying to halt disease progression. It's just because of the complexity of the disease and the typical approach of like taking one drug into the clinic, it's really hard to find the one drug that we think would really halt disease in all patients. And so the goal is finding multitudes of drugs that slow disease progression in patient populations and then combine those and hopefully then really halt disease progression.
Excellent, thank you. We do have one more question that I think is directly relates to your presentation today. So I want to make sure we address that verbally, and then we'll end with this one. Taryn has listed my email in the chat box, so thank you for that. Please email me questions and we will get them all followed up. And yes, I will send an email out for the questions that are presented to me. I will send to all of the participants. So yes, that will be coming. Someone asked, I don't believe my husband was tested for biomarkers when he was diagnosed. Should biomarkers be tested on all ALS patients?
Well, I'm biased that way, so I'm going to say definitely yes. And there's some great reasons they should be, because as I've showed you, you can identify subtypes of the disease that way. And if that's determined early on, that actually might impact the types and approaches of treatment that are provided to the patient at that point in time. So I think it should be included and for all the patients. And I'm pushing to try to do that and to make that a possibility. There's just challenges with reimbursement, who's going to pay for the cost to do all this. So number of challenges that are present, but the goal is to really have that opportunity to provide biomarker feedback on all patients.
Okay, perfect. Thank you, Dr. Bowser.
Sure.
We are going to go ahead and move on because it's time for us to close up. But I did want to say, first of all, thank you, Dr. Bowser, for all of the work that you're doing in the field of ALS research. We really are blessed to have such a hub of research available to us, and we appreciate you always being willing to present and answer our questions. If you haven't already done so, you should be seeing a slide of the CDC ALS registry. If you have not already completed an enrollment with the registry and you are a person with an ALS, this is an important thing for you to do. It collects data from people diagnosed all across the US, and that data is then used for research. So I encourage you to do that if you have not done it. A link to the recording of this session and an evaluation, as well as answers to all of these questions, will be mailed out to you guys next week. And then up next, starting from 11 to 11:45, we have 2 sessions, breakout sessions. Mark Arnold will be joining us to present on inexpensive and readily available technology that can ease everyday life. And Lisa Rayberg will be discussing life insurance policies as an additional funding source for your care needs. So please be sure to check your confirmation email or the reminder text that you're getting, because that gives you the new link. Every session is on a completely separate link. So please make sure you do that. I think that's it for right now. Dr. Bowser, do you have any final words that you'd like, or are we good?
Well, we're good. I just want to thank everyone for participating. Great questions, and I'll get the rest from Amy, and I will answer those for you. And again, just want to thank everyone for participating today, and have a great rest of the day and the weekend.
Thank you.
Bye-bye,
You too. All right, we're going to log off, everyone. I'll see you at the next session, and I am collecting your responses. So